what I'd like to do is introduce Dr. Steve Strauss from Oregon State University. He is another distinguished researcher, has worked on a variety of issues related to the genetics of especially trees. He's going to talk to us about lessons learned from regulated field trials of transgenic trees and implications for potential gene drive applications. Steve, it's good to be with you again, and it's all yours.
Thank you, Jim. Everybody hear me?
Yes, I can.
Great, lovely. Keegan, Audrey, thank you for setting this up. Appreciate it. I'm going to take a pretty different perspective than Mark just took, talking a little more generically, a little more as a practitioner working with plants and trees, so the opposite end of the spectrum with respect to things like life cycle. So what I'm going to do is tell you a little bit about what I've done with transgenic field trials, and then particularly with CRISPR, the status of that, which I think relates to some of the risk, and some regulatory concepts, a little more generic than what you heard from Mark. I'm also going to assume a perspective of something I haven't read a lot about with respect to gene drives, which so far has been very much about how to minimize all possible risks. I'm sort of going to talk more about how to sort of get a balance between the ability to do research, including field research, to innovate and develop technology while keeping risks under control. And I'm sort of one who's sort of got the scars of doing GMO kinds of work with trees, with plants, in the last 10 or 20 years, where it's become extremely difficult to do it. And I think it's possible in coming up with responsible regulations, if we can avoid some of the issues that have come up in the GMO world, that I think would be a good thing to do. So I'm going to come from that perspective. Another piece of broad perspective is in things I've read about gene drive and risk assessment. Sometimes it seems as though people are thinking the world is static and the only risk is from how we might impact it. And in the world that I see in forests and really in all forms of human economy and environment, things are changing rapidly, and where huge problems are occurring, and we need tools to deal with it. So again, we need to have this balance between the ability to innovate and the ability to act responsibly. And oftentimes, unless you can get things in the field and see how they operate, you don't really know what you're doing. And if it becomes extremely expensive to do that, you cut out the public sector, small companies, and you end up with a very, very few players who can do it. So again, my perspective is one that we need a smarter balance. That's not just all about avoiding risk, but it's also about allowing innovation to occur. So a little bit about my background in field trials. So I think in the public sector, I've probably conducted more field trials with GMO trees than anybody else in the United States. I haven't checked the APHIS database yet, but that was true a few years ago. Lots of it was populus, which is poplars, cottonwoods, aspens, the model tree for biotech worldwide. And a lot of it's dealing with flowering modification. So we need to grow the trees long enough that they can flower. We need special containment situations so that we can observe some flowering safely, and that's not easy to do. Probably done about 30 different field experiments. The current experiments you're looking at a few years ago is about 5 hectares in size. Whoop, there we go. Like I said, we look at flowering. We also often want to see what happens to wood, what happens to rates of productivity, what happens to adaptation as we do our perturbations. And so we need to get these trees in the field for a while, usually exceeding what you can do under most grant proposals. So that itself is challenging from an academic point of view. And then, of course, they're monitored, they're authorized by USDA, and it takes several years to actually make the sites so there's no more trees available. Poplars sprout from roots, and killing them is not easy. So it's quite an expensive, difficult commitment to do this kind of work, and I think there's something like 3 laboratories in the U.S., certainly less than 5 that are able to do it, 3 to 5 academic laboratories because of the cost and issues involved. So just to show you a couple of pictures here, another field trial that's ongoing, very pretty. This is sweet gum. We're trying a couple of different containment transgenes, sterility transgenes. This one took about 6 years to just begin flowering. So again, long-term commitment that we try to manage, not easy to do. And then also in the past, we've worked with companies, other researchers to test some of the more well-known transgenes in trees, such as Bt transgenes. You can see one paper below from earlier work. Herbicide tolerance, wood modification, lignin modification in particular to make trees less costly, less environmentally damaging to pulp or to turn into ethanol. So this is the last slide. It's just from a paper we recently published. As you move up, as things get more complex, as you deal with simple traits like herbicide resistance to more complex traits such as the one you see here, modifying plant hormone metabolism to try to improve growth rate, change the allocation of biomass, depending on the objective of what you're growing the trees. We find very much that greenhouse results tell you almost nothing about field research, about field results. Different field results can differ because these trees are interacting with the environment in a complex way. Their physiology changes during age. And so really to do pretty basic research, or basic in the sense of seeing if some of the scientific concepts really apply in a tree, a similar thing would be true for annual crops, you need to get out in the field or you can be very, very, very much misled. And as I said, that's challenging in the U.S., costly. It's much more difficult in many other countries, in fact, to even get permission to do a field trial with GMO trees. And this is not speaking to gene drive at all, just any kind of GMO. So that's very quickly done a lot of that and managed to get permits to do it, and I think I've stayed in good stead, and we'll come back to that in a bit. So what do we do with CRISPRs? This is something pretty new in my laboratory. As you guys know very well, a lot of people are very excited about the technology, say big, bold things like you see in this quote here about it's game-changing. And as you'll see for us, where we're trying to do genetic containment, it really is game-changing for reasons you'll see in a moment. So I think this is not just hyperbole. And then, of course, the interest in using it for gene drive, it and related technologies, people say similar things, and I think it's true. So we need to really pay attention to this and hopefully develop it wisely. So what we do is we work on complete bisexual sterility, male and female, to promote coexistence, to promote regulatory compliance, public approval. And we work on trees. You see poplars here in the upper right, grown in the Pacific Northwest, eucalyptus down below from Brazil. Pines in many parts of the world are grown with this very, very high intensity. Generally, in the most productive plantations, they're vegetatively propagated. What you see in both of these pictures are individual stands that are comprised of single varieties or clones. And so we want wood. We don't want seeds or fruits in the case of most food crops. So the idea of a complete, very simple bisexual sterility is appealing in terms of being able to achieve compliance. So we've been working on this for about 20 years as the different technologies have come along. And so now we have site-directed mutagenesis. We've worked with zinc finger technology and found it, not really very good in our hands, at least. We began to work with talons and then quickly abandoned it in favor of CRISPRs, given their efficiency and ease of production. So we also worked with things like Barnase, a cytotoxin that, in a sense, it ablates reproductive cells, but in our hands, it often causes pleiotropy. can make it difficult to produce transformants, can produce trees that are not as healthy as trees without these. Even floral promoters are not 100% faithful, so that's something you need to be careful about. We work a lot with RNA interference. We have for many years, but we've always been concerned that the suppression that we would get might not be complete enough for compliance or public approval, even if it was for biological purposes, depending on the trait. And of course, if you had a site-directed system that's highly efficient, it would need to produce biallelic mutations, that is, knock out both alleles in our trees. We don't really have the ability to inbreed. Trees get very sick when you inbreed them. They produce a lot of variability. It takes a very long time to do it. And so we don't want to do that. And so one of the amazing things about the current technologies, it seems, as you'll see, very much adequate to producing such knockouts that was really not possible before. Another nice thing is because you're making a physical change, not a change in expression, we really would not like to wait the 5 or 10 years it might take until flowering begins in a field trial to see if it's working well. We'd like to know immediately as we produce a transgenic in a Petri dish, preferably. And of course, if you're changing the sequence, you can do that. It can be very reliable once you learn from research. And we think there are ways, we haven't done this yet, through very, very strong transient expression. Most people who do applied plant biotechnology don't like to use protoplasts. They're very difficult. We're hoping there's ways to avoid them, but still create a mutagenesis rate that's adequate, or to include your CRISPR-Cas system as a transgene, but then knock it out later with recombinases or a similar technology. Right now, as you'll see, we have not worried about that, getting rid of them yet. We've still been showing that it can work. So this is the kind of stuff we've been using, these systems where we can have, as you can see here, I think you can see my cursor, very simply, there's one single guide RNA targeting one particular site in a floral gene. We also have constructs that have 2 of these so far, targeting 2 different sites in a single floral gene to try to get larger mutations, such as deletions. And then a control construct just with Cas9. The genes that we've been targeting are ones that we've worked on for many years, are well known from basic plant biology, are known in poplars pretty well. Leafy, as you see on the left, and in situ showing this expression during inflorescence development. This will be a catkin, this structure. These glowing areas are the places where there are floral primordia. And then also the agamus gene, which you can see expression here again during the developing flower primordia itself, so not the whole catkin, but looking within a flower here, more directed expression. And in the case of agamus, like many genes in poplar and many, many plant species, there's many that are duplicated. There's a recent duplication in poplar where you have thousands of genes with close homologs that both are expressed in different ways. And so prior to CRISPR, we wouldn't even have imagined trying to knock out this gene because it would require knocking out 4 alleles, 2 loci, 2 alleles each. But in the world of CRISPR, as you'll see, that seems quite feasible. So here's the general workflow for us. Very simple, standard leaf disc type transformation, transgenic shoots, and then amplification of DNA from those shoots and sequencing and alignment to determine whether our target sites have been mutated. We're doing similar things at off-target places in the genome, and that data is not in yet to see if there is off-target mutation. In the case of leafy, it's a very unique gene. That's a single copy, which is also true in most plants. We'd be surprised if that was a problem. In the case of agamus, which is a Matchbox gene, that's more of a large worry. So this is just a look at a little bit of data from a PhD student. So what you see here is in yellow is the target site. Here's 2 different target sites in the same gene, the leafy gene, and these are different kinds of mutants that we've seen. With circled are some deletions. We also see small insertions, and with the double construct, we've seen some very large deletions, several hundred base pairs, that all of which will destroy the function of the gene very nicely, we would think. We see down below, many of these are homozygous mutants, where both alleles appear to have the same knockout mutation. So this is a little summary of data as it was about a month ago, and I'll just show you the very, very bottom here. We get about a quarter homozygous knockouts, about a half mosaics, where we have, in most cases, those are two different mutations that are also knockouts, but we also have wild type plus a knockout there. We're still analyzing these. And then in about a quarter of them, we have no mutations. So far in the controls, we've only analyzed 14 events so far. We've seen no mutations to date. So will we think these phenotypes give us interesting, do we think these mutations will give us interesting phenotypes? We don't know yet. It'll be a while for us. We have ways to accelerate flowering in the laboratory that we're doing. We're also trying to get these out in the field. But we've been working on a field trial for a number of years with a lot of RNAi, RNA interference constructs, including leafy and endogamous that are out there. And I'll just show you a little bit of data from that. So here's wild type here to the left as a flowering life cycle. The catkins coming out in March, starting to mature. April, they've been pollinated here, and then releasing seeds in May. And then here's a suppressed leafy mutant. We have two lines like this where the buds really don't develop very far. The catkins are sort of stilted in their development. And we've seen that in two separate years. We also see that when you look at the growth rate of these trees, whether they're controlled or whether they have the mutant flowers, they appear to be normal. The leaves are normal. The trees seem to grow normally, branching normally, normal cycle of dropping leaves and growing in the spring and frost tolerance and so forth. So we're hoping that these are a type of bisexual mutation. So far, we just looked at female trees. The males are just starting to flower in this field trial. But leafy affects both genders. And so we're hoping this is a way to get bisexual containment using CRISPRs without undesired side effects. So hopefully, the CRISPRs that you saw the data on earlier will be ready to put in a similar field planting next year. Hopefully fall, if we can get approval to do that. So the last thing I want to talk about is sort of my concepts of regulatory approaches in a more general way than Mark talked about. So how should CRISPRs be regulated? You know, I think of the question of, in general, how to approach regulation. One approach is to say, well, things moving in the environment can create significant social, economic problems due to adventitious presence, and we've seen a lot of that all over the world. In Oregon, just a little bit of wheat that was left in the environment after field trials of no environmental or food safety consequence almost had a catastrophic consequence for our trade in wheat from the Northwest. So under those kinds of criteria, you might say, you know, we need the strictest containment one can imagine, probably much stricter than we have today in most cases. However, I think from my perspective, the world needs to figure out ways to accept levels of adventitious presence from safe research in the field, from normal commerce, and that will come. That's not going to be easy. And so I'm focusing on sort of biological innovation criteria for what regulation should look like. And also in my concept is that we should be following more of a product, not process concept. Could be more concerned about the traits we're affecting and what their health and environmental consequences could be, positive and negative, less concerned about the process itself, be it GMO or gene drive per se. And in my mind, is we'd like to, if possible, avoid stringent, very simple triggers, for example, that would put every kind of gene drive in the same regulatory tier of complete and absolute containment, because I think that will make it very, very difficult to go through from basic research to application in a timely manner, given the needs we have out there in the world. So I've written about this kind of stuff. I just put these papers up to say these are great papers. You should go read them. About trying to come up with tiers. This is in the GMO world for plants. The paper here, published in 2003. Similar paper published a few months ago. The motivation was tree health, forest health, and the problems we have, and how we'd like to use GMO technology to deal with it, but can't. But it also made recommendations for regulations that were product-based for agriculture, for crops in general. So I'm not going to talk about details today, but there are another level of detail given in these papers if the committee is interested. So I just want to give a little bit of a hopefully somewhat humorous aside, why regulation right, why getting regulation right really matters. Tell you about a funny thing that happened to us, the strange case of the upright summer cactus. I think Sherlock Holmes had an episode on this. So many years ago, we were doing some field trials of semi-dwarfism genes in poplars in the field. In the middle of the summer, amazingly, these catkins, I hope you can see them. I'm pointing at them. I'll show them in a little close-up, started forming in the middle of summer. So catkins don't form on in the middle of summer anywhere in poplars in nature. They're never upright. They're always hanging from branches. So this is very, very strange indeed. So, and here's, I think, a little bit of a closer look at them. There was also a lot of partial conversions of leaves to flowers that were very, very interesting. So biologically, we were fascinated at what was going on and thought this may give us an insight into the action of gibberellins that we were modifying for these trees. But we also knew that under our regulatory permits, we needed to report this to USDA promptly, any unexpected occurrences or a permit condition. So we did that. So we reported it to APHIS, an unexpected occurrence. We discussed with them, well, what do we do about this? It's out there in the field. We had flowering field trials, ones we had permits that were nearby, but this particular trial had not been authorized under that permit. It was sort of done separately. We didn't expect flowering to occur. So, but we wanted to leave it around to study these novel flowers, the expression of the genes, and what was going on. So the risk seemed to be zero, and it would be difficult to remove every one of them from the many dozens of trees that were out there. So we argued the case. I pointed out that this is a very safe. We're dealing with a fitness-reducing gene. We're in the middle of summer when there's no pollen to pollinate. These are female flowers. There's no habitat suitable for poplar in the dry Oregon summers. The seed must move in the spring when it's moist and find bare mineral soil without any shade. Poplar seeds are not dormant. They're not going to sit in the seed bank. And so the APHIS scientists agreed with all my arguments, but they looked at the legalities and said, well, you know, we think technically this is a breach of your permit condition, so we need to report it to the compliance branch of APHIS, which, as far as I could tell, was some kind of police force that prosecutes bad guys. And so that got me very concerned. This is sort of how I felt in terms of what the next steps might be about this. So I'm trying to be a little humorous here. I hope it's working for you. I bet I can hear you hopefully chuckling a bit. And I had images of this kind of thing happening. And you may know that in the U.S., at least, permits from APHIS, from USDA, they're your personal permit. They're not institutional permits. So I indeed would have been personally responsible. So, well, luckily I heard that this was happening, and I decided that rather than risk whatever might happen, I had no idea, rather than risk what might be publicized embarrassment, what might be called disregard for the environment, who knows what kinds of institutional complications might have occurred. we went out there, got every student in my lab to get out there and remove every single cactus, and did the same the next year when this happened, and this is sort of what it looked like. This is the next spring as they were forming every individual tree in the trial, being carefully examined, demonstrating to APHIS that everything had been removed and autoclaved, and that was the end of that. But the reason I go through this little thing is to talk about science versus law. It was a bit of a wake-up for me. This could have had very, very serious implications for my career. And I realized that, you know, it didn't matter what science arguments I had. It didn't really matter what the scientists at the regulatory agencies thought. It was the letter of the law that mattered, and that our current system really does assume in practice that use of the GE method makes something potentially very dangerous until you can prove otherwise, and that's simply the way it is. And, you know, as I said, biology, safety, benefit are irrelevant in those laws. So had the rules been written based on product rather than process, the trees would probably not have been regulated at all. A trait's safe. It has economic and ecological value in its use. And the risk from a field trial release, from the extremely small size of the field trial, no compatible wild relatives in the nearby area, make it extremely safe. But that was not the case. So there was great risk to me, to the institution, due to this very, very small violation. So not quite done with a little story. One answer we had is, well, you know, we want to study flowering in trees like this and our other trees. Why don't we just deregulate them just so we can do good research over many, many years without all these risks of these small compliance errors? Of course, to do that, we need to deregulate several constructs we want to study, several insertion events, to do good science, to compare them, to see the relationship of gene expression to phenotype and so forth. So I did actually go to APHIS, and many contacts with them, and suggested we just deregulate them as a category. And although I've always found the regulators that I've worked with to be extremely helpful, in a sense, this was the reaction, not out of not being nasty, but just that you can't do that. So each event needs a pile of data. They need to protect themselves from from lawsuits, to withstand NEPA type of lawsuits. Basically, they need something not that different from an environmental impact statement for each event. And of course, doing this requires years of research. We're trying to get some way to do the research affordably and safely. And so, in a sense, what we really needed to do is to just provide a mountain of paperwork. The question is, do we want the same for all of our CRISPR and gene drive applications? So the bottom line is, from my point of view, is it's critical to make regulations about risk and benefit and not about the method per se. In the world that I'd like to see not just gigantic corporations able to use the technology, but public sectors and small company type of operators. And each product should not have billion-dollar profits in order to justify it. So we're not there in the world of GMOs, and I hope we don't get there in the world of CRISPRs and gene drive. So then how should we regulate? How should regulations for gene drives intended to solve plant problems be structured under a system like this? So just some ideas for you. So what are some targets we think could be done with a high level of safety during field research and application? So I'm not the first person to say these. These have been published in different places. I'm repeating things. Herbicide tolerant weeds, when the drive is focused on new or now prevalent resistance alleles. So these would be things that, because we have an herbicide that has to be used, these presumably would not have a noxious effect in the wild environment should they get out there. I think I also meant to say exotic plants as well. Damaging invasive plants that don't have compatible wild relatives. We have lots of those in Oregon and elsewhere that we have no real economic means to control. Damaging exotic insect pests and pathogens, such as those that are damaging wild and cultivated trees. I'll show you a couple of examples of those in a moment. And the question we'd ask ourselves, do we need strict regulation of field trials for any of these cases? Are these cases where we might provide exemptions after an initial review, or registration but not full regulation, is that thinkable in the world of gene drives? And it seems that perhaps it should be. So the kinds of pest problems I'm thinking about, I think about trees. Those are the examples. I'm sure you all know about the case of citrus greening. That is an existential threat to our citrus industry in the United States. And indeed, it's probably too late to save these citruses given the slowness of either GMO or gene drive approaches under the regulations we have and the state of science. Other things I think about a lot is talking about forest trees as compared to horticultural trees. There's lots and lots of issues with the health of forests around the world, so much so that Science actually devoted an issue to that in August, which is, I think, the first time it's ever done that. And these are due to exotic pests moving around. These are due to changes in climate, making native pests much more serious problems. And so there's lots of cases where I can imagine gene drive being a great way to perhaps help combat an exotic pest. You probably all know the case of chestnut blight, a common wild tree on the East Coast that was extirpated as a tree by the blight, and I could imagine gene drive being a very powerful way possibly to combat that. It would have certainly been very powerful had we been able to use it early on in the spread of the blight. Hemlock in the USA under siege from an insect, now spreading. You can see this map down here, killing large trees. Very, very difficult to think about dealing with without a technology like gene drive or biological control. Emerald ash borer spreading throughout the United States. As far as I know, all or virtually all ashes of many different species are susceptible with a direct cost in the billions of dollars, actually much larger than that if you count the ecological and amenity costs. Again, would seem like perhaps a good candidate for gene drive if it was done aggressively. So what are some of the mitigating factors in my mind that would give you more versus less safety in research and applications? Obviously, the size of release is very, very critical. The location of release, as Mark talked about with the mosquitoes, of course, very, very critical. But even a small release in an area where there could be some uptake, I would guess would be safe in many cases, as to have a large impact, particularly with organisms with a longer life cycle, would take massive and repeated releases to really have an ecological effect within many decades. So obviously, if we do have a deactivation system that's been produced by a developer and made publicly available, should something go wrong, that could be applied in an aggressive manner to reduce or reverse the problem. And of course, gene drives that powerfully suppress fitness. And the case I'm working on with CRISPRs, where we hope reproduce completely sexually sterile trees, would be an example that probably innately are extremely safe. If that was combined with some kind of partial sterility with some kind of gene drive, that would also make it much safer, of course. And then in general, when we're doing gene drives that are mainly inserting within existing genes, so we're producing loss of function kinds of traits, we know those are very, very common in nature as mutations, natural mutations. And so on a gene-by-gene basis, we could think about whether those are really a threat or not, ecological threat, beyond what's in the wild. So what kind of things require more scrutiny and care for responsible research? Obviously, when we're dealing with essential genes and we have ecologically compatible and, most importantly, common wild relatives, we need to be concerned. When we have damaging invasive plants, again, where we're focusing on important genes where there are wild relatives there's broad sexual compatibility and they have important ecological functions. And of course, damaging exotic insects and pathogens that also have ecologically important wild relatives that are carrying out other functions. I think these have all been said before. So in summary, again, the perspective I've taken to go, I think, a different way than most of the writing I've seen, is that regulations, the burden can be huge. The burden for genetic engineering in general, in my view, is extremely large and has taken a large part of our innovative capacity in the country and the world out of innovation. And in fact, the Obama administration in their call for revised regulations said exactly that. So we need a smarter way to regulate. And I'm hoping that even though in the early National Research Council reports, this is what they said, strongly in fact said several times, as have other scientific societies, that we should have a product, not process focus, it really has not been put into practice hardly at all, in the sense of regulatory tiers, both during research and in terms of regulatory approvals. And again, to emphasize, how we come up with regulatory triggers is critical, because in the world we inhabit today, the baggage of being labeled a GMO or a gene drive or a CRISPR is very difficult to recover from. And so if we don't need to do that in some way, we should think about that seriously. So as I've said, I think tiers like that seem feasible even in the world of gene drives. So I hope the committee will give that serious thought. And I just want to thank the people in my lab who've done the CRISPR work, who've done the field work, and the sources of support that I have gotten for this kind of work. Thank you very much.